time.
Sure would be.
That would be amazing.
We may actually, I'm hoping that just this video and this site might stimulate some groups to actually get very interested in trying to do that.
Mm-hmm.
It's no easy task to get a current profiler that's got the pressure housing
Yeah.
down at these depths is extremely expensive, I think. Definitely is not like, hey, let's go out and put a current profiler down. No, it's not quite like that.
Yeah, being able to come back out, being able to deploy it, being able to pick it up and retrieve it again. Any of those kinds of methods, I mean, it always provides such amazing, results like tracking of large animals and seabirds and things like that. But the actual process of deploying those instruments and getting them back, getting the data off of them is quite a project.
And thank you to all of our viewers out there. Thank you for all of the questions. I just spot them and I ask them. I got a very nice message from one of our viewers, and please write your names as well with your comments so we know who you are and where you are viewing us from. So yeah, the questions that I asked, those are the questions from our viewers. So thank you. We have another question here about sponges.
Yay! What's that?
How deep, how long do they survive without food?
Without food?
Without food, do they have any energy food storage organs?
I can't answer that, no.
It's a great question.
Yeah, it's a great question. So I don't think they have any way to store food because they're a very simple organism that doesn't have well-defined organs whatsoever. It's more almost a collection of cells, specialized cells.
Mm-hmm.
And so if the cells are deprived of a food source, if you will, then I don't think they're going to be able to last that long.
Mm-hmm.
So it's a very good question.
And we had another question earlier. How do sponges grow? Do they have growing planes?
Another good question. No, these are really good.
These are very, yeah, anatomical, physiological, chemical questions.
Well, there's so many different shapes and sizes and types of sponges out there that I think there's multiple ways that they grow. For example, we're coming up on a leaf sponge right now, I believe. And this particular sponge is going to be dividing, but it's also going to be secreting a lot of silica. And of course, this one's going to be growing upward. It's going to be growing small, and then it starts growing out these leaves very slowly. A polyopigon, those gigantic, huge monster sponges, I don't know. I mean, they all got to start small, and presumably that type of sponge is growing relatively symmetrically. It's adding material and cells and creating the silica spicules more symmetrically, both laterally and vertically, or in three axes, X, Y, Z, because it's also got some girth to it. I'm afraid that's the best I can do. Very good question, and I'm sorry I don't have a better answer for you.
Well, one of the best things about this organization, Ocean Exploration Trust, is that whatever our scientists are watching now live, the whole world has the opportunity to watch and observe and discover new things as we are. So thank you for all of the support to continue this great work, understanding our oceans, observing, surveying the ocean floor, trying to understand the systems that support all the living things here.
That's a pretty magnificent leaf sponge we're coming up on right now. This is in the genus Tritopleura, but we call them leaf sponges because they look like leaves.
And it looks like it has the black corals growing on it.
It's either black corals or crinoids. What is that?
Crinoids.
We could zoom in on them. Let's find out.
Could be hydrozoans as well.
Go ahead.
This one, I'm not seeing that black coral. I think that's more of a hydrozoan. Yeah, so I don't think this one has that commensal black coral, that Silospongia, which is currently known as Anapathy.
Oh, wow, that's very clear.
Yeah.
Can we get some shots of that, Canelo? Thank you so much. Oh,
Little shrimp.
that's the sediment that I keep seeing. This one is not full of it, but it's very interesting.
Yeah.
Very crisp images.
Yeah, great shot. Thank you very much.
It's really getting rattled, isn't it?
Yeah.
Viewer trying from Denmark is wondering if all of our 2021 live streams are available to watch later. Does anybody know?
Bet Aaron could answer that.
Not the whole dives. They'll put out highlight reels and different highlight clips, but not the entire dives.
And those are in our YouTube channel, right?
Yep.
Yeah, thank you, Aaron.
This thing lost its head. It's a dead stalk, probably a Caulophacus, I would assume. Got a lot of hydrozoans or hydroids growing all over it, squat lobster. And so even when these sponges are dead, they still serve a purpose, and they're still helping the community because they're providing a way for a lot of other organisms to get up into the water column.
Crinoid there.
Yeah, crinoid. That's Cyathometra. It's a beautiful little crinoid. There's an Umbellapathes, that little flower-like thing down there. It's another type of black coral. Boy, at this site, or right here at this site, black corals rule.
Yeah.
Let's see. Okay, so somebody asked earlier about the deepest black coral, and Tina, who specializes this is Tina Molodtsova at the Shirshov Institute in Moscow. She said the deepest one that she knows was in an Okeanos dive in 2017, which was at 5,772 meters.
Whoa.
And it was genus Abyssopathes. And this is a big Corbitellinae new genus is all we know this guy from. And we sampled a smaller one of this. So that is a nice size one of those.
Mm-hmm.
But black corals actually get down to trench depths, frankly. And they call depths, I think, below the normal level of sea level, which is, seafloor. 5,000 meters is usually considered to be pretty much the base ocean depth. And then you've got these trenches, this is 5,772. And once you get below about 5,000 meters, then they call it the Hadal zone. So black corals are actually getting down to the hadal, which is
Sure.
remarkable.
Sounds good.
Well, we're getting a lot of questions here, so we have plenty to answer during our blue water.
Oh, good.
Yeah.
Yeah, definitely this site has stimulated all the scientists and the public alike. It's quite a incredible community we have. Chris is scheduled to put us on.
The team has put us on.
The team has put us on, yes. Very dynamic. So much to see, so many different animals.
Yeah, and I'm saying team because, first of all, you can't even dive on a site like this unless it's mapped first. And the mapping was initially done by the Okeanos program in 2009. And then Erin and her team here mapped portions of this as well. And then it's got to be processed. There's John Smith downstairs who's processing the data and making the HIPAC displays possible.
Fish.
It's quite a collective effort. Yeah, let's get the fish.
Let's get the fish.
Let's get it.
Let's get them.
Get it. Bump it, slurp.
Oh, no.
As you can see, the lasers are running right through it, and it doesn't bother them at all.
Looks like it's snake, doesn't it?
It's going to be Iliophis again. They tend to be more slender synaphobranchids.
Oh, backing up.
This is the second one we see on this watch.
Oh, I'm sorry. That was a big move.
Yep, I can see the white lateral line pore, so this is Iliophis. It's a genus. I don't know what species this is, but
Off into the abyss.
okay, so now we're
Nice.
coming
Oh,
around.
I love those sponges.
Oh
Those look
yeah,
really
if we
cool.
could take a look at this guy.
Yes,
this
we
sponge.
can.
Mm.
I don't know. That's a contender
Go ahead.
for my favorite.
Zoom,
I love the
please.
form of those.
Okay, I'm pretty sure this is in the family Phareidae, and I think it's in the genus Pharia. I wonder if we could get even a closer zoom if possible. I want to show you something really cool if we can.
Ooh, ah.
It's a little bit too old. The tissue's a little thick here, but this has a really recognizable framework that looks just like a screen door. You know, it's very symmetrically the way the spicules are all lined up. This is definitely a Pharia, and we call this one Pharia oca erecta because there's two forms of this. There's sort of a low bushy form, which is the Pharia oca, and I guess we're calling it Pharia near oca, and then there's this taller version that we call the erect form of it. So beautiful sponge, very thin-walled. They're very brittle as well, and we've sampled this before. Spectacular. Thank you very much.
Thank you.
All right, we're going to try bearing change 220.
Roger.
Bridge nav, can we get 20 meters 220?
By the way, I'm not necessarily finding a dead ringer for the photo. I'm going for the actual name.
I think we have better
multiple
photos.
growth. Yeah, I just knew that this one had the name on it. So
Got it. Thank you.
Connor is noting that some of those magnificent sponges we saw at the beginning of this particular dive are in contention for some of the largest sponges ever found. And I would agree completely, Connor. Those I've never seen, these polyopicons so big. They're just magnificent. And they're probably contenders for the oldest sponge in the world too, but we just don't know how old these things are. But it's got to take a while to get that big.
Long while.
We have a viewer from Istanbul.
Huh.
Oh, wow.
Yeah.
Cool.
Welcome.
Aloha.
And his question is, why don't we see parasitic organisms? Wouldn't it be a good idea
That's a great
to
question.
parasitize other organisms in food-deprived areas?
We do see parasitic organisms here. We have seen fish that have copepod types of parasites on them as well. We just haven't seen any today. Let me think of some other examples.
Well, there's just infinite examples, except we don't know what all the relationships are that are going on between these organisms that are living on the sponges and living in the corals, whether they're parasitic or symbiotic or commensal or, right? I mean, we don't know a lot about how it affects their metabolism or growth, because we don't know a lot about the...
I guess I'm trying to remember the official definition of a parasite. So it doesn't
lives inside
necessarily
or
kill
lives
its
on.
host,
Yeah.
does it? But it's also harming its host in some manner. And so it's not predation. It's not like a sea star eating a coral. And so I know that the fish parasites that are down here is one example of it, the copepods. I've seen these relationships between sponges and anemones and hydrozoans, but they don't seem to harm each other. It's more of a commensalism.
What about that? Isn't there was one other one that you mentioned where it actually took over the skeleton of a black coral?
Oh, that's true.
Or a gold coral, right?
A gold coral, but we haven't seen any at these depths. That's a species. That's Kulamana homau. Boy, I'm going to say that wrong. Haumea, excuse me. That's around 400 to 500 meters depth, and we're quite a bit lower than that.
Yeah.
That would be probably a parasitic relationship.
Yeah, I would assume taken over.
That's a really good question. So thank you for that. Again, our answer is a little lame, but we did the best we could.
There's a doctoral thesis in there, I'm sure. What do you got, 15 minutes, Aaron? What are we looking at for?
Yeah, 15 minutes. I think it will get us to the surface probably right at 8, but I think a little late is better than a little early for Mark's breakfast and happiness for the rest of the day.
Happiness. I like that.
You mentioned a cocoa pod. That reminds me. That sounds like a nice warm drink on a cold winter day.
A copepod?
Oh, a cocoa pod, not a cocoa pod.
A cocoa pod? Oh, look at it.
A cocoa pod.
Wow.
I wonder if we could take a quick zoom on the sponge. I think we've seen it before, but...
The left gray one or the white
left
fluffy?
Larger one, sorry. This guy right there.
Yeah, okay.
They could be the same sponge, just different young one and an older one. This is really another Faria. This is...
Oh.
Okay, zoom in, please.
That looks pretty rosy.
Oh, okay. This is a very old vase sponge, euplectellid vase sponge. So this is in that Corbitellinae new genus, if you will, but it's about the oldest one I think I've ever seen, and I'm calling it old because of the lengths of these protrusions coming out of the exterior surface of the sponge are longer, I think, than I've ever seen for this type of vase.
Little shrimp peering out from the inside.
Yeah, it's almost trying to grow a sieve plate over the osculum right on top, which is pretty weird.
And what is the scientific name of this sponge?
Well, we don't have a species or a genus name to this thing yet, if I'm identifying it correctly. We just
know... And no sample today?
No, yeah, it's similar to, what we sampled already yesterday, I think.
Oh.
I'm going to call it Jester's hat. A Jester hat.
What do we have here? Oh,
leaf sponge.
Leaf sponge. Okay, thank you.
These black corals in the bottom just remind me of fields of seagrass, you know, in shallow waters.
Would you say that this leaf sponge also looks like a cactus?
It could, you're right.
That's what one of our viewers.
Yeah.
Like a Joshua tree.
I wouldn't have thought that until he mentioned it.
Yeah.
Thanks for now affecting my thoughts about this sponge from now on.
And Andy, yesterday you noticed a sponge that looked like a sombrero.
Yeah, I did, yeah.
Those giant sponges.
We were talking to your class. Yeah.
Well, I'm going to stick to leaf rather than cactus.
Kale leaf, please.
Kale, it does look
Kale
like
leaf,
kale.
that's right.
Yes, it has that frilly structure.
And Jason, I don't know if this is our usual Jason, he's asking, how are some sea stars able to eat glass sponges? Are they eating silica, spicules?
It's a good question. There is and it probably is Jason because he's seen this before. We haven't seen this today, other cases, but there's a sea star that's in the genus Pythonaster that eats sponges, and somehow it's able to digest the tissue and then deal with the silica spicules in its stomach. And I'm pretty sure that Chris Ma of the Smithsonian out proved that they were eating sponges because he actually examined the guts of this particular, some collections from this genus, and they found spicules in the stomach content.
Really?
So they must be able to get rid of them or shed them somehow, but they're,
Maybe they're using them as digestive roughage.
I don't
like
know.
Fiber?
Fiber, yeah. Enhance their digestive processes.
Maybe, but that's really specialized. But then you think about the sea stars that eat the bamboo corals. The bamboo corals have very spiky sclerites as well, and they have to be able to deal with those very sharp, hard objects as well.
I was also reading that some of these sponges can also use aragonite for their structure. I didn't know that.
Yeah. That's going to be the demisponges, I think, which we did see some earlier.
Those are those like half cone structures, low to the ground, wide attachment point. Yeah.
So when Professor Kelly says old sponge, how old does he mean? 1,000 years old or so?
Yeah, it's some of those sponges we saw last watch were so enormous it would not surprise me at all if a few of those could be 1,000 years or older. But we really don't know. This is a new emerging field, being able to age the sponge using radioisotopes, I think.
When do you think we'll be able to know?
Well,
tomorrow.
There are some researchers out there that are really working on this and are really trying to come up with some solid methods to do that. So it's going to happen. It's just we're in the embryonic stages of that field of research. But it's a total guess right now based on the size. That's all.
And what's the biggest size of sponge that you think we saw yesterday?
Last night.
Oh my God.
Last night we said that.
Pilot, what do you think about that? Our biggest polyopogon.
Oh,
like half a herc.
Yeah, 2 meters wide, 3. I don't know.
Yeah, they were.
Have to review the tapes.
And
yeah,
thick.
They were just huge.
They were a meter thick, some of them.
Yeah.
So speaking of age, is the deep sea equivalent of older than dirt in the deep sea is older than sediment?
Yeah.
I'm surprised you two got any sleep last night. You were so excited.
I didn't. I was dreaming sponges and giant coral communities.
I will admit that I took an Ambien. I apologize. I needed some
You were
chemical
not
help.
the only one.
I am so jealous.
You were vibrating
You were
last
not
night,
the only
Chris.
one.
Oh my gosh.
On nights
Sounds
like that.
The best night ever.
I know.
Yeah.
But they hardly work because I woke up before my alarm.
Yeah.
And I was like, oh my gosh, did I miss my watch?
No, I think I went to sleep around midnight. I just couldn't stop watching it and just wanted to see more and more.
Connor was asking about the black coral spawn. Connor, we didn't really notice anything in the water column when we got on watch this morning. We were further down on the slope, and the water looked pretty clear. So, yeah, I think somebody's going to have to look at the videotape and try to confirm this possibility that you were suggesting.
Well, one thing they noticed, Chris, and I saw this before I went to sleep, was they some of the black corals up top, they were looking at the bases, and Steve was commenting that it looked like potentially like either a brooding. It was actually in a reproductive stage because there was this little bulbous, clear bulbous part near the bottom of the black coral that may have been brooding larvae or producing larvae. So there was some physical manifestation that also triggered that observation, and I don't know enough to speak to it, but we'll have to talk to Steve when he's up.
Quite frankly, there's two types of, there's probably more, but there's two types of reproduction in corals, and that's either releasing the eggs and sperm, and you have external fertilization in the water column, and then the subsequent development into planula larvae is what their larval stage is called, that actually will settle out eventually on the substrate. And then the other form is sort of more of an internal or an on-coral type of fertilization. There's a sea star, please, if we can zoom in on it. And so they actually release competent larvae. I was just trying to use the mouse to circle the sea star. That's
not going to work for you.
No, it wasn't really working for me.
You wait till I zoom in, and then you can circle it and make the target
bigger.
Okay,
you need to be a little tighter before I can circle it.
Yeah. Okay, zoom in there, please.
Yeah, it's this one. Sorry.
That leg? Okay.
And this has got something on it, it has one of those little pill bug looking things.
Oh. Yeah, it's probably a polychaete, maybe.
Uh-huh.
Ah! What a ride.
I think it's a goniasterid. Let's leave it at that family. I'm not positive what it is. But
all right, thanks.
What I was saying is the other type of ways coral spawn is there's sort of this fertilization before they release the larvae, and the larvae actually develop on the coral or in the coral itself, and then the coral releases the competent larvae right away, and that larvae can settle immediately. And I don't know if that's what Steve was seeing or whether he saw some eggs, because I didn't see the observation.
I'll have to grab the screen grabs, and you can take a look at it, see what you think.
Oh, I can't get over there.
What a compliment. Competent larvae.
Because right now we're
competent larvae.
Yeah, we're at the head of this little knoll on the ridge, and so maybe we're going to get into some bigger things.
Oh, but we're not, because we're coming up.
5 minutes.
Yeah, that's okay.
-5. There's that sponge that or dead sponge to the left of that.
Yeah.
Polyepigon. Down in the little sediment area there.
Since we are in the last couple minutes, we've put in the last ship move. We'll let everything settle out. Is there are we niskinning or anything?
Oh, ooh. I'm sorry. Yeah, we probably should do a niskin. Thank you very much for the reminder, Aaron.
Thank you.
Yeah, so these are water samples that we take at depth, and we can...
Are we doing one like right now?
Yeah, let's go ahead and do it if we got... I think we're far enough up, aren't we? I think she wanted it 15
like
meters?
10 to 15.
You wanted to do one like 5 to 10 meters up bottom, is that right?
Or 10 to 15, I think
10 to 15, yeah.
I guess, Trevor, whenever you think it's appropriate to take that, we'll do it.
Sure. Yeah, we can do it right now. It's very appropriate.
Okay.
And I'll just come up a little bit. You're good there, Antonella.
Well, that was sure 16 hours of spectacle. Really, really amazing.
Yeah, I think
as soon as we as soon as we hit bottom.
Yeah, by far, this dive has been just constant
Yeah.
amazement of life
Shocking.
in the deep sea.
Just so gorgeous. Just like an ancient, walking into an ancient forest, quite literally.
It is really.
You got 234, I think.
Yep, 234 is open.
So these water samples will be analyzed for environmental DNA,
Sure.
eDNA.
Yeah, yellow, blue, orange.
Which is a way of looking at what's in the water column based upon the genetic material that's floating around, what's living in this area.
So our typical dive is about 12 hours, right?
This
We
one
had-
was, this is 16,
This one was 16 and our longest was
24, I think.
24.
Yeah, part of the reason we opted for this shorter dive strategy on many of the seamounts is because we had to do a lot of rock collection, both for Dr. Orcutt and for Dr. Balbus here for aging and for the microbes, and we also needed some biology. So in a sense, we needed to clear those boxes.
There
do
you
a
go.
shorter
Oh.
Uh-huh.
So that was
Yep.
part of the reason why we did this. Longer dives, once you filled your boxes, you really can't collect any more items there. Got it. Oh. So close.
There you go. Yeah, that's it.
She's got it now.
Yep, got it.
Perfect. Way to go, Antonella.
Nice.
Yay.
Nice, thank you. Was that number 3?
Okay, we'll get set up for recovery now.
Well, Andy is...
That was blue? Yeah, that was 3, yeah.
3, thank you.
Oh.
And that was sample number 159.
Well, all good things have to come to an end, folks, so we're reluctantly...
Okay, shall we do the ceremony?
Yes, we
please.
must. We must be very thankful to the waters of Kanaloa. So to all of our viewers out there, we are ending our dive, and this is a very special protocol that we do. So we are going to go radio silent to conduct our private ceremonial protocol to acknowledge and honor the biocultural resources that are present and that we are interacting with and impacting. We will be back shortly on SPL.
Sorry, we're gonna do this one twice just so everyone, so we know. Okay. Mahalo.
All right. We're back.
Okay, let's start coming up.
Oh, wow. The adrenaline's all gone.
Wow.
It's
After this wave of experiences, my dreams will never be the same.
You're going to be dreaming of massive sponges filtering you.
Uh-huh.
Yeah,
That'll be
no,
your night.
I know.
I wonder what it feels
just
like to be
horrible.
filtered.
Mm-hmm.
Oh, I had dreams of these giant rocks before, but now
So we got some time here for some questions. Let's go for it.
Yeah.
What do you got?
Okay. We'll start with the, I think what requires shorter answers, and then we'll go to the longest answer questions. Um, hmm. One of the questions is, would the sand particles in sandy patches be the same age as the rocks? Or is the sand still sinking from shallower reefs?
I don't think the sand is necessarily coming from shallower reefs because there are no shallower reefs around us or near us. So sand is coming from someplace else, and that lighter colored sand is probably organic in a way. It could be diatoms or pteropods or even decomposing coral branches. There may be silica spicules in there. So some other source would be my guess, and therefore it would probably be younger by quite a bit than the surrounding rocks. However, they did notice on the previous watch that they were finding manganese-coated what appeared to be sediment. And it's possible that the sediment inside those rocks was quite old in order. Sometimes manganese crust will actually precipitate on sediment, and to the extent where it can form a coating, and once it's got a film over the sediment, then it can start building on that to actually create these type of features. So though that type of sand could be quite old.
Okay. Smarties. Oh, wow.
You'll spread these out. Thank you very much. There you go. A couple for you. He's gotten two for everybody.
That's nice. Thank you, Trevor.
Yeah, thank you, Trevor.
And
what's our next one?
I think we're ready for the voices of our ROV pilots. We have a couple of questions here. We know you have been very focused controlling our ROVs, and so we'll start with, how are the ROV Hercules and Argus controlled?
Well, I'll talk about Herc first. Herc has 2 joysticks for motion underwater. One of them does forward, back, left, right, and turn left, turn right. And the other one does up and down. And then there's, of course, automatic controls too, our auto, which we don't really use unless we're holding position, but it keeps us in one spot more or less. Those are kind of the 2 ways we move Hercules around.
For Argus has a lot fewer controls than Herc. We can control the vertical or depth with the winch, so we winch up and down. It also has 2 thrusters, which we can use to turn. We could use it to go side to side, but we don't usually do that. We just change the heading around, so usually to look at where Herc is.
Oh, thank you, Antonella and Trevor. Another question is about a way to house electrical equipment. This viewer is wondering if inexpensively sealed enclosures filled with a mineral oil will be able to handle those depths, since the oil behaves as an incompressible fluid.
Good question. I'm going to come right back to that after just a little operational discussion. Stand by.
Okay, thank you.
Okay. I heard there was a question on electrical blah blah blahs. Yeah. Okay. So, Herc has and Argus have two ways we can use electricity in the ocean. First one is for dumb stuff like just wires, just basic wiring. The only that can handle pressure, no problem. But the only real thing we need to consider is keeping the water out. So I think the viewer mentioned mineral oil, and that is absolutely correct. We put them in a what's called a junction box. Wires come in, wires go out. We connect them in the oil. And as the ocean squeezes onto those wires, the oil pushes back with the same pressure and keeps all the water out. Now the fancier stuff, the smart stuff like computers and sensors, etc., those don't want to be wet, but they also don't want to be squeezed. All the silicone chips have little bits of air in them and they would implode. So we keep those in pressure housings. These are usually titanium or stainless steel, milled out of one big chunk of metal and designed to withstand the pressure of the ocean, which is quite a lot.
Wow, thank you for those answers, Trevor.
Sorry, what was that question?
Yeah, I think you answered the question.
Oh, great.
Thank
okay.
you.
Thanks.
Hmm. Okay. Thank you. Well, thank you, Chris. So this is very exciting to read where our viewers are watching us from. We have Giovanna from Brazil. She's 17 years old. She wishes to make marine exploration her career of the future. And she says that we are the reason this childhood passion came back to life. Do you guys have any advice on how to make it a reality? So she's 17 in Brazil. And
okay. Well, I guess the only advice I can give you is take your education very seriously, get the best grades you possibly can, and also definitely consider getting into college. I think she's going to be about 1 year before she's ready for college. And once she's got into college, again, take that education very seriously. If you want marine exploration, you need to determine what area of marine exploration really interests you. Is it geology? Is it the technology and the engineering that goes into it? Is it the biology? is it the mapping, what area that really she's quite interested in, and then just go for it. You know, there are options to actually get some hands-on training. This program here, the Ocean Exploration Trust and the Nautilus provides internships here that'll give you hands-on training. And that's how a lot of people out here started, because everybody gets a chance to look at you and see your performance and so forth. And then that could definitely lead to some job opportunities out here. So that would be the best advice I could give her right now.
Yeah, thank you. Thank you for that advice. And I'm sure other viewers out there are asking the same question. And no matter how old you are, you can always work on a new passion, right?
That's right.
When did you start studying sponges, Chris?
When did I start studying sponges? Well, I began as a fish guy in aquaculture, believe it or not.
And you began as a sponge.
Yes. Well, I was looking at fishes until I got the job at the Hawaii Undersea Research Laboratory, where I had to learn a lot more animals. I had to expand my horizons a little bit because that was the nature of the job. And then as we were doing that and we're trying to create this database and animal guides and everything else, the group that was very poorly understood, the most poorly understood of all the animal groups that we had and we had recorded over the years, were sponges. And so I just decided that I need to know more about them. We've got to improve on how we're identifying sponges and how we're recording them. And so I just buckled down and started educating myself about sponges. I was meeting and interacting with world's authorities and people who really specialize in sponges, trying to learn as much as I could from them. I had a wonderful opportunity years ago to actually go up and spend 2 weeks with really one of 2 world's authorities on sponges and live in his house and work with him in his basement.
Wow.
And I've been to animal identification workshops at the Monterey Bay Aquarium Research Institute years ago, where I met a lot of people, including sponge experts. You just go for it.
Yeah.
That's
How
it.
old were you?
I was older. I was in my 40s, I would get, late 40s or so, because I only started working for the Hawaii Undersea Research Laboratory in 2000. So that was 21 years ago. So,
yeah, I asked that because many viewers think, oh, I'm too late to start learning about ocean exploration, but it's never too late.
It is never too late.
We have viewers in their 60s, 70s learning about it. And,
yeah, I'm a bit of a late bloomer myself. I didn't get my doctorate until I was about 40 years old. I used to be very, very interested in tennis, and I'll give you sort of an analogy of that. I used to give tennis lessons quite a lot in summers when I come back from college. And that was a way to help not get out of college with too much debt. And anyway, I just remember this one guy, and he was taking up tennis, and he was 80 years old, and he was taking up the sport, and he could beat a lot of the younger players as well that were there. He was learning so quickly.
Wow.
It is never too late. Just pursue what interests you as best you can, and just go for it.
Yeah, thank you. Thank you for that insight. I guess this is a question for everybody. Do you have any recommendations for books on deep ocean creatures?
No, but I was going to add on to the thing about advice for careers. Go check out the page with all the profiles of the team members and find the career that you're interested in. Find those those team members and look up what they did to get there, because everyone has like their universities listed, their education listed, their experiences and jobs before listed. And that's just a really good way to see, like, these are the people like with the jobs I want, how did they get there? And that's a great way to figure out your own path.
Yes, that's an excellent point, Erin.
Yeah, thank you very much for that.
Yeah.
Great.
I don't
And
have any recommendations for deep sea books. That I don't have any help.
I was wondering, there is a book, and I'm trying to remember what the author's name is. I met her. I think it's called is it called The Abyss or something like this? It's a big, huge coffee table book. It's got a lot of pictures in it with some really
Sylvia
good ones.
Earle is one of her books.
Sylvia Earle, you know, that's very early books.
There's
that
a new one called The Brilliant Abyss by Helen Scales, that S-C-A-L-E-S, that I was just about to start, and it's one of the more current books about deep sea exploration, and also I think it covers some deep sea mining stuff, so it looks in that budding field, so it looks kind of interesting.
I think Dr. Ballard also did a book, and maybe co-authored with somebody else, on exploration of deep sea wrecks and things like that, if I remember. And I have that book. I'm just sort of forgetting right now.
I love all the works by Callum Roberts. He does an incredible job at covering the hundreds of years of ocean, humans in the ocean and exploration. And I also read a fantastic book recently by Enrique Sala. He has a book. He's an ocean National Geographic explorer in residence, and the title of that book, it was so wonderful. But in talking about careers, he really went into his life and his passion for the ocean and how that developed and how he got to where he is today. That was a really wonderful ocean book. I'll find the title here in a second.
yeah, thank you.
Yeah, it's called The Nature of Nature by Enrique Sala, E-N-R-I-C Sala. And he is, again, an explorer in residence, National Geographic ocean explorer in residence. And it's just a wonderful, wonderful book.
Thank you, Andy. And Chris, how about you, Kainalu? Do you have a favorite ocean-related book?
Yeah, just a moment. I think I found the book that I was thinking of. Let me see. Trying to Google it right now.
I'm not too sure.
Yes,
I don't
okay.
know too much deep sea ocean books.
So this, I was trying to
love these
remember her name. In 2007, Claire Nubian was the author of a book called The Deep, The Extraordinary Creatures of the Abyss. And I have that book, and I met with Claire. She came out to Hawaii after that book came out. And if you want to see some spectacular imagery of deep sea creatures, and they've got some text describing a lot of these animals, it's a really, really neat book.
Thank you.
And it's not that expensive. So if you wanted a new one, it's $45, and they also have some used ones for $9.39. But sometimes I just flip through that book because she's got a number of animals that I've never seen in there that are really beautiful, full-page images.
I'm going to have to get that for my library in my classroom now.
Yeah, that's it. Claire Nubian is the author.
Or also, Aaron, you didn't mention a book, but do you have any Instagram pages or podcasts or any?
I would just say if you're interested in ocean exploration, find all the different programs that do it. So follow Nautilus Live, follow NOAA, follow all of the ones that'll give you content and resources.
Yeah, all these three, well, the three that come to mind off the top of my head certainly are OET Nautilus, and then you also have Schmidt Ocean Institute that also broadcasts their explorations, and then NOAA Ocean Exploration on the wonderful vessel, the Okeanos Explorer. And all three of these entities together have recently come together and put up a wonderful ocean education site, and they share a lot of their resources, and it's a one-stop portal for all the different expeditions that are going on, and I'll see if I can get the URL for that here. But that's a great one-stop shop for education materials, find out if there are any live dives going on for these 3 different entities, and lots of other resources.
Mm-hmm. And would you mention Seabed 2032?
Yeah, well, Aaron probably can do a much better job about explaining that, Seabed 2030, than I can, given that that's...
Yeah. Aaron in navigation?
Yep, sorry.
We're wondering if you could, talk about Seabed 2030.
Oh, I would love to talk about Seabed 2030. So Seabed 2030 is an initiative to map the world's oceans by 2030. And as we've talked about a few times, only about 20% of the world's oceans are mapped to any kind of high level of resolution. And for us, mapped means mapped with a modern multi-beam system. So Seabed 2030 is funded by the Nippon Foundation. They also fund the JEBCO program, which is the Generic Bathymetric Chart of the Oceans. And they train scholars at University of New Hampshire and University of Southern Mississippi to become ocean mappers and take their skills back to their country of origin. But anyway, so Seabed 2030 is partially part of JEBCO, but also its own standalone program. And there's regional data centers set up to first and foremost, do the data mining and find out where there is data we don't know about. So they've been working on that for quite a few years now at the different regional data centers. And then to do what they can to support ongoing mapping efforts to try and get this continuous map available. So the data is, the database for Seabed 2030 is in, Boulder. And it's IHO kind of runs the whole thing, but it's the, oh gosh, what is it? The name of the database, the DCDB. Sorry, can't remember what the acronym is, but they're bringing all the data together, organizing it. And so for us, for instance, we collect multi-beam data specifically over certain areas, but anytime we're on transit, like our transit out to this site or even between seamounts, if we see a gap in the existing bathymetry, we do our parts to try to fill it in. And then our data goes to Lamont Doherty. And then from there, it gets distributed to NCEI, which is NOAA's repository, but it also gets sent to the appropriate Seabed 2030 regional data center to try and build up that grid. So yeah, it's just this effort, and personally not sure that we could possibly achieve the goal, but it's really exciting to have a goal and have people excited about the initiative and trying their best to contribute.
Wow,
Yeah.
Thank you. That's a great...
Thank you, Aaron.
Yep, no problem.
And a couple of our viewers were wondering how that mapping of the ocean floor is done with ROVs or ships. Can you explain the basics of that?
Yeah, well, I mean,
Thank
it could be done with either or others as well. So we do our mapping from the ship. We have a hull-mounted EM32 multi-beam sonar. That is a 30 kilohertz sonar, so we can't quite get to the deepest ocean depths like the Marianas Trench, but it does cover the majority of depths that we work in. And so we use acoustics to map the ocean. So the multi-beam sends out an acoustic pulse, many acoustic pulses, and then it returns back, and we know from the time it took to send that pulse out and for it to return, we can then use the speed of sound to calculate the depth of the seafloor at that location. And so that's kind of the basics of all ocean mapping. It's almost all with acoustics. In really shallow water, you can use laser, LiDAR data. You have to have pretty clear water for that to work. And so that's how we do it, but you can put a multi-beam on whatever you want. So we've had a multi-beam on Herc. We